Liberty, which is helpful for me, is they transferred all that stuff that went into from the hunter to this card. And this is what we use to track that sample. Okay. From here, once we get the sample, we put this all into a spreadsheet, a database, and then we track it that way, and then we communicate with both our lab and the Colorado State University lab on what samples we're submitting, so we're all on the same page as to what the results are. So it's a pretty good tracking system. What we try to collect is a tooth, a genetic sample for potential future research if there's ever demonstrated that there's maybe some resistance to this disease, or if there's some genetic aspect to this, we'll have a sample on storage so that basically forever, so that we can go back and do some additional research if we need to. And then there's two bags, and these are the bags for the lymph nodes. Again, they're all labeled and marked, so we don't have to do any of that ahead of time. It's all done ahead of time. Set that aside. And then we use these knives, a scalpel blade. You gotta be a little careful. These things are really, really sharp. Pops in there, so we're good to go. Forceps. And what I like to do is try to keep my workstation fairly clean. A lot of people want to set the head on the cutting board. That's probably not the best thing. I want to put my samples there. So I'm trying to make sure, so somebody talked about, you mentioned cross-contamination. Yeah. So once we get the sample out, that's where I start worrying more about that kind of stuff. So I don't want to put samples from one deer in the same spot. If I'm doing multiple, I might divide this board up into halves or fours and put different nodes there so I can keep it all separate and then clean it off in between. The idea is to try to be as efficient as possible. You know, it's not bad now, but when you start getting hundreds of samples in, you got to be fairly quick at it, otherwise it'll take us forever.
Neil, can I ask you a question while you're doing that? You had indicated that cross-contamination can happen at a water source or a collective feeding site. Have we been able to rule out airborne respiratory cross-contamination? You mentioned feces,
yeah,
and I'm wondering if it dries out and becomes airborne, if that's a possibility.
So far, there's no evidence of that. You know, what we do know is it can be environmental contamination. So, say this deer is defecating, urinating on the ground, and it has prions in there, in the feces and the urine. That can contaminate that site. If this animal is to go off and die somewhere and has CWD, those prions will persist in there, and that can get into the soil. And there's research that suggests that plant matter can actually maybe take these prions up into the plant material.
Oh boy. So
you're a deer, you come by, the site's contaminated, you eat it, now you have a potential new source of CWD. That's one of the reasons why this disease is almost impossible right now to eradicate. So eradication is not really something you can do. It's unfortunate, but it's just the way it is. It's a very hardy thing. It's a protein. It's not a bacteria, it's not a virus, it's different than what most people think of in terms of disease. So our body produces proteins normally. It's the normal process. But those proteins are also broken down by our body. With CWD and some of these other diseases, these prion diseases, that prion takes a different fold, and the body doesn't seem to recognize it, it doesn't break it down. And then they start to accumulate. And the body also sees that, and there's evidence that suggests that it starts to replicate those misfolded proteins. So they get a small dose could cause a pretty major infection in these animals because the body starts to replicate them. And then when they appear in the brain, they'll cause damage to the brain. So it's called chronic wasting disease because the condition of the animal starts to deteriorate over time. And at the end of the stage of that disease, they're nothing but skin and bones.
How long does that take?
It can take 16 months to 2 years.
Is there any evidence that there are some animals that are immune to it?
There's not, well, I shouldn't say, there's one case that we know of, but there are some genetic strains of deer and elk that seem to have, I don't want to use the word resistance, They die from it, but they die at a slower rate. But they are very much underrepresented in the normal population. So they were selected against for some other reasons, which nobody really knows why, but for whatever reason, they're not real prevalent in the population. It might take them 2.5 years to die rather than 2. There's one case of an animal, an elk, I think they named her Lucky, but in the research facilities in Sebille, Wyoming, where they first found and documented CWD, highly contaminated site. They can't keep elk alive long enough in there to do any other kind of research. There's one animal that's been alive for 8 years, and they have no idea why.
Well, we
So-
need to know why.
Yeah, it's the only
That's
one though,
important.
so, if you're looking at that as a hope, one out of all the different, I don't know how many hundreds of animals they filtered through there, that's not good odds. So yeah, there's no known immunity or resistance.
So total eradication of a herd doesn't get rid of the prions, they're still there. If you reintroduced new
Yep.
animals, they get sick too.
Yep, they tried to disinfect those facilities and they weren't successful to stick new animals in. You got a new case. All that said, we still think the primary mean of transmission is through animal-to-animal contact.
For the 10 cases that you have, do you know at what stage they are or how long they've had CWD?
We don't. We can learn a little bit from the age, and the symptomatic ones were probably at the end stage, so they've had it for a while. But an animal like this could look perfectly healthy to us, and it could be in those early stages. You could still shed those prions, and you wouldn't know it. And that's what makes detecting this a challenge, because there's no live animal test. Hunter shows up at a check station, we can't look at this deer and say that's CWD. We gotta seize that. You have to take the lymph nodes out, you have to test it, all that stuff takes time.
So that's 16 to 24 months, that's once they start showing?
That's
So
from the time they get CWD.
From the time they
It
contract.
takes they live for about 2 years after they contract the disease.
They can do a lot of spreading in that
And
2
they can spread it
years.
basically the whole time. So this is pretty typical. This is a hunter harvested sample, so we'll take the sample. And so those lymph nodes are located right back here. And so there's a lot of different ways of doing it, and I'll try to, if you guys want to look, you can look closer, but want to cut a little bit closer here. And just get that exposed so I can see the esophagus. And you can see the neck. And if you look, you can see there's a little bit of connective tissue right here, right between the esophagus and the muscle. And that's where I want to go. I'll try to. And you see, I'm just keep cutting down here. Open that up, and I actually nicked one of them, which isn't a big deal. And so here's the lymph nodes right here. You can see them. And they look a little bit different. So you have salivary gland right here, which is this kind of globular tissue. And for the folks that aren't used to collecting them, sometimes they grab that instead, and that's the wrong tissue. What we're looking for are these lymph nodes. And they're a little bit different texture than the muscle. And you can see I kind of cut one right there. But they reside right in little pockets right. Here's the jawbone, the mandible. There's little 2 pockets here right behind the palate. And we want to collect those. Put that on my. And the other one is right there. And that's about how big they are. They're about the size of a large kidney bean, if you will, or depending on how big your thumb is, I guess about the size of your thumbnail. And those are what we're sampled. On deer and elk, that's the primary sample. On moose, we take the brain. I can show you where that's at, our brain stem. So you want to find that joint right there. You see how that neck moves? And I want to get access to the spinal column. So if I split it a little bit, and there's the atlas and axis joint, And it sits like this, so the vertebrae come over the base of the skull like that, with little wings, so it's a joint, so it can move. And you can see them right here. Here's this part of it, and there's the skull part of it. And if I just hit that joint just right, I can open that up, and it reveals the brain, the spinal column. Now this one may have been frozen, which makes it more of a challenge. We'll see if we can get it out. It might be a little bit mushy, I can tell by the way it moved. So that's why fresh is important. The longer you freeze these, or the more you do to them, the harder it is to get those out. But you can go in with your knife, and go around, cut it loose. And she's got these little grape spoon. These things, these aren't my favorite, but Jennifer likes them, so she brought them. But I like a little spoon, but you can kind of go in behind there, and we'll see if I can get it out. You can see how mushy it is. So this is the challenging part, which is I'm glad we don't have to do this in all of them.
Just moose?
Just moose. Yep. Yeah, I'm not
Why,
gonna get this
why
one.
only for moose?
So,
What's the difference?
just thinking I'm gonna have to get this out without making a mess of it. Yep. It's just too, yeah, it's too pliable.
Yeah.
Yep, and it came out.
Does that have any value though? Right
It
there?
doesn't right now. I was hoping I'd get it, but yeah, I think this one is frozen. And you can see how soft it is. It's just mush. So that's why it's hard to get out. So the question is why different for moose? And these animals, those proteins or those prions are distributed through the body a little bit differently in different species. They all get it, but how it shows up in these tissues here differs a little bit between deer, elk, and moose. And for moose, what we want to do is we want to be able to look at the brain as well as the lymph nodes. Plus, we don't have as many samples for moose, so they're trying to get a better idea of exactly how it manifests in moose. So they want both samples, and they run a different test. So when we first test these, they'll run what they call an ELISA on it, and basically they'll grind it up, and they're looking for those proteins, and they'll test for those proteins. So if you get a hit on that, they call it a suspect. And then they'll take another piece and they'll run a second test. You know, histochemistry, where they actually take a slice of it, they stain it, they mount it on a slide, and if it has those proteins, it kind of fluoresces, and they see it. And when we see that, that's like the gold standard for the test. And so we've got a double check there, and that's a confirmed positive. So we run 2 tests on any of the positives, just to confirm that it truly is. The reality is, when it comes back positive on ELISA, I don't think they've had a case yet that hasn't come back positive on IHC on the second test.
Is there a false positive on the lymph node? I mean, like, is there a possibility that any of
Well, that's why they're doing the second test, but it's doesn't seem to happen. I mean, they're both pretty good tests. So what we are doing too, then, we're splitting these samples about 50-50. Is a little off. We'll send a bigger piece to the lab. Okay, and then 1/2 will go to Colorado State University, and we bank the other half for if you want to do any future research on it or surveillance. So we've got a sample kind of in the bank.
Did you ask him your question you had about deer to elk?
Oh, yeah. What sort of research has been done on showing the transfer of the disease from species to species, from deer to elk or elk to moose?
You know, there's a lot of the research is a little bit more anecdotal in that where you find it in deer, you typically can find it in elk if you look hard enough. There is some evidence that, yeah, cervid to cervid transmission is possible, but you can actually go in and they can identify, now they're getting sensitive enough they can actually identify what species this might have come out of based on the type of prion. In the past, they didn't have that technology. So there's definitely I think there's cross-species transmission between the cervid family, but once you get out of there, this prion does not jump species barriers very well. And that's why they don't think it's not caused any human disease that we know of to date. So you imagine Colorado, Wisconsin, Wyoming, where they've had this since the 60s that they've been able to document. Hundreds, if not thousands, of people have probably eaten CWD-infected animals, and yet we've had no human cases. But even though it doesn't jump species barriers very easily, that precaution is, hey, if you know it's positive, we still recommend you don't eat it because things happen. Mad cow disease is a very similar disease to this. We didn't believe it would jump the species barrier, and then we found a few cases in UK where folks, it did cross that barrier.
So when you say jumping species, an area has infected deer is not necessarily going to infect elk or moose.
I think there's potential. Those are so much more closely related
Okay.
that there does seem like it can move between.
What about livestock, though?
Livestock, no. So it's a disease of deer, elk, moose, and reindeer. The cervid family.
Strictly.
Yep. So far,
So you got whitetails that have tested positive. How could it go from them to the mule deer through that plant scenario? you were
that's another
talking about?
reason. But there's interaction between these deer on the landscape. I mean, just because they're different species, sometimes we have crossbreeding. Sometimes they're in the same area. There could be just some normal interactions. And especially if they share a food source where there's urine, feces, that type of stuff, the mule deer comes in, eats that. There's another route. Same with the elk.
Can the prions be spread by, let's say, a horse grazing on plant life? Go through the digestive